All right, hello, welcome to the segment of Chapter 15 dealing with linked genes and linkage map. Now, sex-linked genes, so the genes that are located on the sex chromosomes are considered sex-linked genes or linked genes. And linked genes in general exhibit different patterns of inheritance than seen before. And we kind of saw that with the eye color in the flies on the first segment. Okay, so the pattern of inheritance is kind of odd, and it's because of the segregation and the fact that the males have only one X. Now, inheritance of linked or X-linked genes, right? So these are going to be genes that are located on the X chromosome. Now, this is going to, a gene, again, remember, can either be on a sex chromosome or not on a sex chromosome. So when it is on a sex chromosome, it's called a sex-linked gene in general. So an X-linked gene, not sex-linked gene, but an X-linked gene is one that's going to be on the X chromosome. Now, genes on the Y chromosome are called Y-linked genes. There are not a whole lot of these. There is what's called the SRY region on the Y chromosome in humans, and that's what gives males or those with a Y chromosome the male characteristics. Now, genes on the X chromosome are called X-linked genes, as we already discussed. Now, X chromosomes, again, they have genes for many different characters. So the X chromosome, remember, is quite large as compared to the Y chromosome. Remember, the Y chromosome has a very small region that is homologous to the X chromosome. So not much recombination, if any, goes on between the X chromosome and the Y chromosome. And the Y chromosome, the majority of it is that SRY region. Now, again, the SRY region codes for sex determination in male. Now, the X-linked genes are going to follow some unique patterns of inheritance. Like I said, that any sex-linked gene is going to show that. Now, for a recessive gene or allele, right, that's on the X chromosome, okay, for it to be expressed, the female, remember this is recessive and females have two X chromosomes, the female must have both copies of that recessive allele. It acts like an autosome. Okay, so it's very similar to the other chromosomes. However, a male needs only one copy because he only has one of the X chromosomes. So it's not, there's not another one to mask that recessive condition. Now, because males have one copy of an X chromosome, they're termed hemizygous. Okay, so it's called hemizygous. Now, X-linked recessive disorders are going to be found mostly in males, and we saw that kind of with the flies, even though it wasn't a recessive disorder, it was just a mutant phenotype, which is on the X chromosome, and it's found mostly in males. Well, the same thing is going to happen even in humans with disorders. Those are going to be shown in mostly males. So it's because males only have one X chromosome. And if there is an allele that results in some sort of condition, right, or disorder, it's going to definitely be expressed. Okay, it may not be expressed in females. It will be expressed if the females have that recessive allele on both chromosomes. Let's look at a couple crosses and how they are, all the genotypes are denoted. So here we can see a female. The female has two large X chromosomes, and then the superscripts show basically the genotype for that particular disorder. Now here we're just using normal versus affected, and affected could mean whatever recessive disorder that we're dealing with. And it may not be a disorder or a disease. It could just be something that is on the X chromosome that really has little effect on the organism in general. So here we have a couple crosses. So we have a female with two normal alleles, and then we have a male that's affected. Okay, so his box is going to be shaded. If we put this in a pedigree, then you would see that. And so we have this cross right here, and you can see you just bring down the x's, and then these x's across here, bring down the y's, and then these x's across here. And then we can see that here, this cross results in 100% of the females being carriers. Okay, so they're carriers because they're heterozygous. So you could call them heterozygous, and you could call them carriers. Now, the males are all normal because remember, they obtained their X chromosome from the female, and the female had both both chromosomes were normal in terms of this allele. Okay, so we can see that cross rare. Now, there's a number of questions that can be generated from this. So it's important for you to read the question carefully, because I could ask, what is the probability that their next child is going to be a carrier? Okay, so if I say child, then you want to count or include both the males and the females. So the probability of their next child being a carrier is going to be 0.5. Now, if I ask you, what is the probability that their next son is going to be normal? Then you would say 1, because 100% of the males are normal. Now let's look at another cross. So we can see here we have a female that is a carrier that is mating with a normal male. Okay, so we do the cross. And again, this is really where it becomes important. So if I ask you, what is the probability of their next son being affected, then you would say 0.5. Because half of the males, or basically the probability, right? So 1 out of 2 possible genotypes can be generated, okay? And one of those is affected, one of them is normal. So if I ask you, what is the probability that the next child is going to be normal, okay, then you would say really 0.75, because this individual is normal, but if I do say carrier, then it only refers to the females. The males cannot be carriers because they can't carry 2 alleles. They can only have 1 allele, and whatever that allele is, it's expressed. All right, let's look at a final cross. Here we've got a female, which is a carrier, and mates with an affected male. Okay, so now we have the results here of the cross. And you can see that 50% probability that the female is going to be a carrier, and a 50% probability that their next child will be affected, right? It doesn't matter whether it's a male or female. Now, in terms of females, again, it's 50%, and then if you separated it in terms of females and males, it would still be 50% because it's 1 female and 1 male. Both can be affected in this particular cross. All right, so that's an example. I'm going to go ahead and do a couple examples just on the writing board so that you will have a variety of examples. And we'll see some actual disorders. Now, some disorders are caused by recessive alleles that are on the X chromosome, and these are some of those examples, and we'll see some in the examples that I provide in a separate video. Now, X inactivation happens in female mammals. So let's look at the cats, because it's most obvious in the cats. So here, remember, the females have 2 X chromosomes. One of the 2 is going to be inactivated in each of the cells. Okay. Now, if a female is heterozygous, she will turn out to be a mosaic. Okay. So this is most obvious in the cats, right? So whenever you see a calico cat, okay, it's going to be a female because that female is a mosaic. So it depends on which X chromosome is inactivated. And the inactivated X chromosome is called the bar body. So you're gonna see what's called a bar body, which is very condensed chromosome inside the cells. All right, so let's look at these a little bit more in detail. Again, It only happens in females because the X chromosome does not become inactivated in males. So here we have a male with the black allele, it's going to be black. A male with the orange allele, it's going to be orange. Now we have a female in which the X chromosome has the one that's been inactivated is the orange one in all of her cells, right? And then this one is a female in which the chromosome that was inactivated was the one with the black allele, so that the orange allele is expressed in all of the cells. Now you have a mixture, right? So these cells, the cells that are orange, the chromosome with the black allele has been inactivated. And in the cells with the black color, the orange chromosome was inactivated. Now, looking at the chromosomal level, we can kind of see what's happening. So this represents a cell in which it's a heterozygote, right? So it has the allele for orange and the allele for black. Okay, so early on during embryonic development, when cell division occurs, one of the X chromosomes becomes inactivated. Okay, so here, if the X chromosome with the orange allele is inactivated, then it's going to have a black fur in those cells. And in the cells in which the chromosome that has the black allele was inactivated, then it's going to have the orange fur. All right, so let's talk about linked genes. And we're going to kind of relate this to Morgan's work with flies again. Okay, so linked genes tend to be inherited together. So they're passed on from one generation to the next together. And Morgan saw this when he was crossing flies, looking at two different traits, so a dihybrid type cross. So again, just a little background, each chromosome has hundreds or 1,000 genes along the length of it, of course, except for the Y chromosome. The genes located on the same chromosome tend to be inherited together because that chromosome is going to be passed from one generation to the next. However, if crossing over occurs, they may not be inherited together, right? So remember, crossing over can occur, but it doesn't occur everywhere. So the majority of the time, a lot of the genes that are going to be on the same chromosome are going to be linked. They're going to be inherited together. So again, Morgan did some experiments with the fruit flies, and he looked at two characters. Right? So let's go ahead and look at this experiment. Now, the two characters that he looked at were body color and wing size or shape. Okay, so let's look at the cross looking at these two characters. So the two characters are, again, the wing size and the body color. So there are two body colors. We've got the gray body color and the black body color. The black is the mutant and the gray is the normal. And then the wing size, we have the normal wings, which are quite large, and then vestigial wings, which are truncated. They're very small, shriveled up wings. And the small wings are going to be the mutant, where the larger wings are going to be the normal. So, of course, we're going to test to see inheritance, so we want to do a test cross and see what's going on. So, of course, you want to take an individual that is homozygous. What your results should be is the F1 should be all normal, right? Because they're heterozygous, and each one of those alleles is going to show the at least one of the normal alleles, and then of course one of the mutant alleles. Now, what actually happened was he ended up with an interesting ratio. So if the genes were located on different chromosomes, okay, then there would be a 1 to 1 to 1 to 1 ratio. But if the genes are located on the same chromosome and the parental alleles are inherited together, then you would see a 1-to-1 and 0-to-0 ratio. And with these numbers, you can see, so this would be basically 2-to-2 to 1-to-1, which is like a 1-to-1 to 0-to-0 ratio. So this suggests that the genes are located on the same chromosome and they are inherited together based on his results. Now, based on these experiments, he found that the body color and the wing size are typically inherited together in specific combinations, and this would be the parental phenotypes. So he noted that these genes do not sort independently, so they don't follow that law of independent assortment, and reasoned that they were probably on the same chromosome. Now this figure shows that cross. Basically, we have the F1 dihybrid female um, and the homozygous recessive male with the cross, and then of course the results. And what the results were basically the parental phenotypes. So we have most of the offspring were going to be either the wild type, which is gray with normal wings, or black with the vestigial wings. It's the same as the parental phenotype. So um, this is what led him to believe that these genes must be on the same chromosome because they're inherited together. If they were not inherited together, you would see different combinations of each of the. Now let's talk about genetic recombination and how it relates to linkage. Okay, so Mendel and Morgan, their studies in genetics kind of relates to the, both of them can kind of overlap and relate to the chromosomal basis of recombination. So remember recombination or crossing over that occurs during meiosis? However, non-parental phenotypes were also produced. So what is going on? What I just described, basically, that recombination must have occurred in those that had the non-parental phenotype. So understanding this result kind of means you really need to explore genetic recombination. Okay, so remember, recombination is basically the production of offspring that have different combinations of the traits from the parent, but different combinations of the parent, so they look phenotypically different. So remember Mendel's P's, he saw that 9 to 3 to 3 to 1 ratio when they independently assorted, right, during that dihybrid cross. So you saw different combinations of the parents. Remember, the parental were homozygous for yellow and round and homozygous for green wrinkled, but then they produced different combinations like yellow wrinkled and green round, right? So this signifies that the genes are either on separate chromosomes or far apart enough on the chromosome where they are segregated independently, because crossing over would occur on the chromosomes in which they existed if they were on. Now, remember crossing over. Okay, so homologous chromosomes line up. They create this tetrad by the synapsis. And then they're going to cross over and exchange equal segments. So then you get more combinations, right? So you can have different combinations of alleles. Now, crossing over doesn't always occur. And it's going to occur on generally different chromosomes at some point, and it's relatively random. Now, mapping, so genetic or gene mapping is basically mapping the distance between the genes based on data from recombination frequencies. Now, Sturtevant was one of Morgan's students. He actually constructed a genetic map of the genes on the chromosomes for the fruit flies. Okay, so this is basically a map showing the location of each gene, their relative locations of each gene on the chromosomes. Now, Sturtevant basically predicted that the further apart the genes are, the higher the probability of crossing over that will occur between them. And then this would basically result in a higher recombination frequency. Okay, so further apart they are, more crossing over, increased frequency of recombination. It kind of makes sense. Now there's okay, so a linkage map is the map that was produced. Okay, and the linkage map is produced by calculating recombination frequencies. Now, this is how by basically determining the distance between the genes. Okay, so the distance between the genes are called centimorgans. Okay, now map units are another name for it. So one map unit is equal to 1 centimorgan, which can be related to 1% recombination frequency. Okay, so the further they are apart, let's say that's 5 map units, which is equal to 5 centimorgans, which is equal to 5% recombination frequency. So the higher the frequency of recombination, the more likely, right? So recombination is basically crossing over. The more likely the genes are going to appear that they are not linked, because crossing over will allow the mixing of the alleles. Now, again, map units basically can be related to the distance and the order, but not the precise actual locations of the genes. That must be. Now, again, this kind of shows a pictorial representation of what I just said. Okay, so here we have 3 different genes. Okay, now you can see the distances between these first of the black and the orange is 9% recombination frequency, which is equal to 9 centimorgans, which is equal to 9 map units. Same here, 9.5. And then we have 17% recombination frequency. Now 9 plus 9.5 doesn't equal 17%. Right? So remember, these are relative locations, and it's calculated by recombination frequency, not using a ruler. Okay, so it's based on the frequency in which crossing over occurs between the genes. Okay, so there's going to be more crossing over between the black one and the blue one than there is going to be between the black one and the orange one, and the orange one and the blue one. All right, so this represents the particular the phenotypes and the relative locations of the genes that code for those phenotypes on a chromosome. So you can see this is basically a genetic map, a linkage map of a chromosome found in flies. Okay, so we can see these are appendages on the head, aristae. Okay, so they could be short or long. And then we have this one right here. We have the color of the body, which is either black or gray. And then this one would be the, they have the cinnabar eyes, which is more like an orange color versus the red eyes. And then of course there's the white eyes as well. And then we have the wing size. So we have the vestigial wings or the long wings. And then we could have brown eyes as well versus red eyes. Remember, red eyes are normal. Brown eyes and cinnabar eyes are the mutant phenotypes, and also white eyes are mutant. Now, in summary, I kind of talked about the sex chromosome, kind of reminding you of those and relating it to Morgan's work, and of course, the assortment of genes on the sex chromosomes. We did a couple Punnett, or I reviewed a couple Punnett squares using genes, right, or alleles from those genes that are on the sex chromosomes. And talked about linked genes, right? So again, those genes that are on the sex chromosome are considered linked. And what is X inactivation? And how does it relate to the sex chromosomes, or the X chromosome specifically? And we talked about recombination frequency, and how it relates to linkage maps